already for this challenge because it is somewhat overwhelming. I'm going to get through it. And here's the great thing about these lectures is we are starting first with the endocrine system, which will be one of 2 chapters covered on the first exam. The endocrine system works in conjunction with the nervous system to maintain homeostasis. I know your topic, your favorite topic, mine too. We're going to go ahead and review it a little bit here. But I want you to take a second and think about what homeostasis is. Go ahead and wait for my computer to catch up here. Got a lot of things running in the background. Think it's going to be a little slow, but we'll see how it works. I'm using an old tablet to record these recordings, as you recall from the welcome lecture, because it allows me to write on the board quite nicely. Hopefully, you can read it. So let's go ahead and make use of that here with a review of our definition of homeostasis, which hopefully you recall is the body's ability to maintain a relatively stable internal environment within physiological limits. Okay, now key things to remember about this: relatively stable. If you're not going to die if your body deviates from 98.6. Okay, you have a high and a low, and if you stay between that high and the low, your body's happy. Now, those limits, that high, that low, those limits can vary depending on which system we're talking about, which homeostatic balance we're specifically referring to. Blood glucose can be quite broad, from 70 to 110, I believe. So it's a pretty broad range of those levels. Blood pH, on the other hand, is 7.35 to 7.45, very, very narrow. So there's always a range, but the broadness of that range will vary depending on what system we're talking about. Finally, one thing you kind of want to get used to is this idea of what is our internal environment. Well, the primary component to our internal environment is going to be your body fluids. All those compartments and the fluids within them. And again, not just the water per se, but all of the solutes as well. All the different components to that body fluid, the solutes, the different mechanisms that those solutes maintain, all need to be kept within specific levels. And that's not only the chemical characteristics, but also the physical characteristics, as far as the temperature, the specific gravity, the osmolarity, all that stuff comes into play when we talk about regulating our internal environment and our body fluids. Now, we're going to start with a little bit of a review. We're going to start with a little bit of a review because the process of maintaining homeostasis is handled by both the nervous system and the endocrine system. And we talked about the nervous system last semester, and here we're going to talk about how it differs from the endocrine system, which is going to be the goal of this semester in this chapter. So, the nervous system maintained homeostasis by sending signals to an effector organ. Now, this effector organ was a muscle or gland. Okay, it was restricted to that. Now, the nervous system detected a problem. That problem was interpreted, a fix was decided upon, and a nerve impulse or action potential was sent down to a neuron to that effector organ. But technically the action potential didn't cause a change. The action potential had to release a neurotransmitter. The neurotransmitter, as you hopefully remember, is simply a type of chemical. Now, even that neurotransmitter alone was not enough to cause a change, because that neurotransmitter had to bind to receptors on our target cells. It wasn't until that neurotransmitter bound to its specific receptors that a change resulted, or some action was taken. Now, quick things to note about this. The speed at which it takes the nervous system to cause a change. What do you think? Is that going to be fast or slow? Well, hopefully you're thinking fast. Action potentials occur within milliseconds, nanoseconds. Not only that, the neurotransmitters are released very, very close to, right next to their target. So it takes them even less time to travel across that synaptic cleft and bind to their receptors. So the response time for the nervous system is very fast. But here's a problem. Are the signals from the nervous system meant to last long? Well, think about it. Do you think there's a never-ending supply of neurotransmitters in the end of that axon? Nope. There is not a never-ending supply. And not only that, most neurotransmitters are replenished up here in the cell body, which means it's going to take a while for that neurotransmitter to be replenished and then travel down the axon to the end of it where it can then be released again. So although it has a fast response time, it is only meant to last short periods of time. So good and bad about the functionality of the nervous system. So now let's go ahead and talk about the endocrine system. Well, here, first of all, the target cells for the endocrine system are pretty much anything. Not restricted to a muscle or gland. Anything in the body can respond to signals from hormones. Okay. Now, those target cells respond to hormones, but here's the thing. We have this endocrine gland, which is either told by the nervous system that there's a problem or detects the problem itself and begins to secrete hormones. Those hormones get put into the blood. Actually, technically, they first go into the interstitial fluid, and in most cases from the interstitial fluid into the blood, where they then travel. Now, as they travel through the blood, they eventually reach their target organ and target cells. But here's the thing. It's going to take a little while for that endocrine organ to first detect the problem or be told there's a problem, then synthesize the hormone, then release the hormone, and then for that hormone to eventually get to its target location. So this is going to have a slower response time when compared to the nervous system. Okay, slower response time. But here's the thing. Do you think there's any limit to how long this endocrine system can cause its change? Now, I'm not talking pathology here. I'm not talking, you've got a growth in your tumor or a tumor in your endocrine gland. No, I'm saying there's still a problem. If there's still a maintained problem, is there any limit to what the endocrine system can do? No, because as long as there's still a problem, all we're simply going to do is keep secreting the hormone into the blood, and that hormone is still going to head to the target organ. So it has a much longer time period. Now, remember, we've released this hormone, it's gone into the blood, it's found its target cell. But again, it still can't take effect until it binds to receptors. Now, just like the nervous system, the hormone has to bind to a receptor in order for it to take effect. So it becomes the specific combination of the hormone and its receptor that initiates the eventual action. So both must be available. Now, wrapping this up here, what you should have noticed by now is that these are perfectly opposite of one another. The nervous system has a fast response time, but only lasts for short periods. The endocrine system has a slower response time, but it's meant to last for longer periods of time. Well, hey, don't these guys make perfect pairs when it comes to maintaining our homeostasis? Particularly since some of these physiological limits. Some of these imbalances may take longer to establish. Okay, so this becomes the perfect pair, which is why they're sometimes referred to as the super system, because most homeostatic mechanisms, most homeostatic or physiological limits are going to be maintained by both the endocrine system and the nervous system. And you'll see that here as we go throughout the rest of this semester, looking at how the endocrine system and the nervous system maintain the functionality of our different organ systems. Okay, note, I've just spent a crap load of time on one slide. This is what I was mentioning in your little welcome lecture that I want you to pay attention to. Take notes, listen to what I'm saying, and actively participate. So when I ask you a question, answer it in your head, okay? If you don't get the answer right, make note of it so you can look it up and try and figure it out on your own, or email me, okay? Just kind of want to make that point before we move on. So let's continue with a general overview of the endocrine system and its functionality. So overall, the endocrine system's functions can be grouped into one of 8 general categories. We have extracellular fluid composition. So this means not only the overall fluid or water of our body, but also the solutes that that water contains. So, you know, the electrolytes, glucose, blood pH, all of these things are regulated by hormones. Metabolic activities, human growth hormone, thyroid hormone, etc., all maintaining or regulating the chemical reactions of our body. Our biological clock, when we feel awake, when we feel sleepy, irrelevant of what's going on around us, is hormonally maintained. Contraction of smoothing cardiac muscle can be modified by hormones, both within the heart, increasing or decreasing its activity, but also smooth muscles of your blood vessels to cause vasoconstriction and vasodilation, or the smooth muscles associated with your gastrointestinal tract to regulate digestion. Glandular secretions. So the secretions of a multitude of different glands, including your exocrine glands, like your digestive glands, your sweat glands, but also other endocrine glands are regulated here. Immune functions are also regulated by chemical hormones, and this includes not only your overall immune function, but also specific immune responses as you actually release chemicals that can warn other cells of an infection and kind of close them off, or release chemicals that stimulate the synthesis or production of the immune cells that are best adapt at fighting that particular infection. So a lot of aspects to your immune function is regulated by hormones. Growth and development are also regulated by hormones, and we look at this both on a overall scale from birth to death, but birth to adulthood or puberty, through puberty with your reproductive hormones, and human growth hormone throughout adolescence, but also overall growth and repair that occurs throughout life as we continually repair and replenish bone, etc., or make muscles larger or smaller depending on what is happening in your body. And finally, of course, reproduction, as a variety of hormones regulate the reproductive cycles of both males and females. Now, note, one of the key aspects to the endocrine system is it's actually made to work in very, very low concentrations. Very low concentrations. And there's an important reason for that. When we go back to our first slide here, one of the key things you want to note is since the hormone, the chemical neurotransmitter, is released directly next to its target location, the concentration of our neurotransmitter stays high. That isn't the case down here in the endocrine system, because we're going to create hormones and put them in the blood at site A. It's going to travel through the blood, go everywhere in the body, and some hopefully make it to its target. this pretty much guarantees that the hormone is going to get diluted. If dilution is a guarantee, well, we better make sure that that hormone can still function even though it is diluted. So what we see is we've kind of built into the mechanism of action for these hormones the fact that they can work in diluted situations. So we will see that. We'll see why that is a little later, but the fact that the hormone is diluted or does get diluted in response to its normal mechanism of action being delivered from site A to site B, they need to be able to work in low concentrations in order to function properly. So here comes our first independent study. Remember, we had talked about this, or I believe I had briefly mentioned it in the welcome lecture, but what you're going to see is throughout this lecture, you're going to see questions highlighted in pink. These are independent study questions. Okay, this means you want to read through the section on your own and take your own notes. And this allows me to free up time and spend it on other areas where I feel might be more challenging. So there's your first independent study, and it's, again, anytime you see these pink letters, that's what you're doing on your own. One thing I am going to briefly talk about is this idea that the endocrine system isn't just a collection of 15 organs that make hormones and that's it. The endocrine system is actually quite diverse. It does possess organs or glands that only serve an endocrine function, and we call these your true endocrine glands. And again, these only serve an endocrine function. Which means they secrete hormones and do nothing else. Okay. Now there are 5 of these, and listing them off from superior to inferior, so to speak, we have the pineal gland, pituitary gland, thyroid, parathyroid, and adrenal. These are our 5 true endocrine glands that do nothing but make hormones. But that is definitely not an all-inclusive list. We see that there are additional hormones that come from organs, but these organs have alternate functions. But we say that this is an organ containing endocrine tissue, which means the overall organ's function is something else. For example, the stomach secretes the hormone gastrin, but that's not the job of the stomach. Okay, the job of the stomach is to digest your food, or to aid in the digestion of your food. But it also happens to have a sub-function of secreting the hormone gastrin. Okay, so here we see those organs that contain endocrine tissue are also a subset of the endocrine system. You have quite a few of these. They are listed in your book. I'm going to try to hit as many of them as possible, with starting with your hypothalamus, the heart, thymus, liver, pancreas, stomach, kidneys, skin, placenta, ovary, testes. I want to say I'm missing one other, but I can't think of it because I know there's 12. But go ahead and get that last one from your textbook. Okay, so again, those are organs that contain endocrine tissue, but their primary function is something else. Now remember what we had said. Hormones themselves aren't enough to cause an effect. Just like neurotransmitters alone is not enough to make a cell do something. We need to also have a receptor. In fact, you can actually argue, since the receptor is what actually tells the cell what to do, so the receptor actually signals your cellular change. You might even argue that the receptor is more important than the hormone itself. So receptors themselves extremely important in the functionality. Now, receptors also are what allow a hormone to have multiple functions. Now you might ask yourself, this hormone that we were talking about, let's say we're going to give, thyroid stimulating hormone, right? Thyroid stimulating hormone is our example. I think you guys can kind of guess what thyroid stimulating hormone does. Okay, yeah, it stimulates the thyroid, not too difficult there. Okay, it is released from the anterior pituitary into the blood. Once released into the blood, that thyroid stimulating hormone goes everywhere in the body. But even though it goes everywhere in the body, it only causes a change in your thyroid gland. Well, why is that? Why is a hormone that goes everywhere in the body only causing a change at the thyroid gland? Well, remember, it comes down to these all-important receptors, because there are only receptors for the thyroid stimulating hormone on the thyroid gland. So even though it's going to places like your liver and your ovaries or testes or your kidneys, it's not making any type of change at those locations because those locations do not have receptors. Also, note, receptors do not need to be the same at different locations. There are a variety of hormones that have a very diverse function depending on location. Insulin is a great example. Insulin has different functions at different organ sites. So in your liver, insulin is going to stimulate the formation of glycogen. But in your adipose tissue, it stimulates the formation of triglycerides and lipids to store away all that energy. But how can insulin do two different things? Does the hormone change between the liver and the adipose tissue? No, the hormone stays the same, but what changes is, again, your receptors. This is why I like to argue that your receptors are more important, or at least as important as the hormone. Now, receptors themselves, when we talked about them in reference to the nervous system, were located in one location, and that is the cell membrane. So all neurotransmitter receptors were located within the cell membrane of the target organ. But this kind of made sense because the goal of a neurotransmitter was to cause some kind of electrical change. And since electrical stimuli occur across the cell membrane, it made sense that that's where a receptor would be located. But here in the nervous system, we have the endocrine system, we have a very diverse potential response for the endocrine system and those hormones. As such, we can have more diverse receptor locations. So here, in addition to the cell membrane, we can see that receptors may also be located intracellularly inside the cell, either in the cytosol, floating around in the cytosol and cytoplasm, or within the nucleus. So we have different receptor locations here. And what we're going to see is, depending on our different receptor locations, there's a different potential signaling mechanism that's employed. And we're also going to see, depending on receptor location, that different types of hormones are able to bind to the cell membrane receptors versus our intracellular receptors. Since you kind of got to be able to get into the cell in order to bind to an intracellular receptor. So we'll talk about that in a little bit. Now, recall, like any structure within a cell, it's continually being synthesized and broken down. Let me go ahead and make a quick adjustment here, as something is hidden on the slide. Receptors are continually being synthesized and broken down. They're continually being not really repaired, but broken down and replaced. This synthesis can actually be adjusted dependent on hormone levels. Since we need both the receptor, sorry about that. Since we need both the receptor and the hormone, we can actually target both areas, both the receptor and the hormone. We can target both in terms of a regulation of that hormonal effect. Obviously, if you think, oh, my hormone levels are too high, well, then let me rephrase that. I'm experiencing too much lipolysis. I'm breaking down too many hormones, or too much fat, excuse me. I know, I don't think there's ever that case, but we'll just use that as an example. So I'm breaking down too much fat. Well, I need to get rid of my insulin then. Well, that's one option, but I could also get rid of some of my insulin receptors. Wouldn't that have the same response? because I need both to tell a cell what to do. So we can actually regulate hormone receptor levels in order to have the same effect as regulating hormone levels. Okay, so we see that there are two options. We can upregulate a receptor by increasing their numbers. This is done when we have low hormone levels, and those low hormone levels can be low hormone levels, and let me back up a minute, a lower than normal response. By increasing the number of receptors, we can hopefully get the biggest bang for our buck, so to speak, with the few hormones we have and increase our response. Okay, so that's the goal of upregulation. Downregulation is the opposite, so we're going to decrease the number of receptors. And this occurs when you have higher than normal hormone levels. And as a result, a higher response than you would like. By getting rid of some of the receptors, what you're doing is saying, okay, I may have 50 hormones, but if I only have 5 receptors, my response is going to be decreased. Okay. Now, one of the things I want to draw your attention to, now that we've kind of highlighted the importance of receptors in the function of a hormone, is the idea when we want to talk about hormone abnormalities and hormone pathologies, we have to realize that it may not only be the hormone that's to cause, but it also could be the receptors that are to cause. Or that are the problem, excuse me. So a lot of people think, oh, I've got diabetes, there's something wrong with my pancreas, which, if you didn't know, diabetes mellitus, which is what I'm specifically referring to, is due to a problem with insulin. And a lot of people think, oh, my pancreas isn't working. But here's the thing. It's not just the hormone insulin that's necessary for the function of, but it's also insulin receptors. And insulin receptors are actually at fault for type 2 diabetes, or the diabetes related to individuals that are obese. So what ends up happening in individuals that are obese is they have excessively high blood sugar levels. And not only excessively, but consistently high. Well, as you will see a little later in this lecture, when we talk about insulin signaling, insulin is released in response to high blood sugar levels. Well, if you have consistently high blood sugar levels, and that means you have consistent release of insulin. That consistent release of insulin causes high hormone levels. Well, in response to those high hormone levels, we down-regulate our receptors. Well, with a decreased number of receptors, we have a decreased sensitivity to insulin, and your cells stop responding to it because there aren't any receptors. And it makes you look like you have diabetes. But here's the thing. Once you regulate the blood sugar levels, you then regulate your insulin levels, and once you regulate your insulin levels, all of a sudden your receptors go back to their normal levels. And ooh, diabetes disappears. This is why when you first see the signals that you're an individual who's obese is developing diabetes, you got to jump right on that, because you can actually cure an individual of type 2 diabetes if you catch it soon enough and you stick to a diet and you maintain proper weight and blood sugar levels. Problem is, diabetes can actually, type 2 diabetes mellitus can actually become permanent, because, at the same time we're down-regulating our receptors, if we're constantly releasing insulin, we're kind of putting a lot of pressure on our pancreas. And we've done, 50 years worth of insulin secretion in 5 years. And you can actually wear out the beta cells of your pancreas, which are what secrete insulin, and that actually creates a permanent diabetic state, as you can't repair those. So this is just highlighting how when we talk about abnormalities in the endocrine system, we have to focus not only on the hormone, but also the receptor. Bringing us to another classification for hormones, or another way of describing hormones, is the idea that you have circulating as well as local hormones. And as the name implies, circulating hormones are those that circulate through the blood. Nothing too tricky there. They are secreted at one location, enter the interstitial fluid, then into the blood, where they travel or are distributed throughout the body to their targets. Okay, remember with circulating hormones, though, dilution is a likely problem. Local hormones, on the other hand, stay local. They enter the interstitial fluid and find their target without entering the blood. In general, there are two groups of local hormones. They're either paracrine hormones or autocrine hormones. Now, paracrine hormones, as the name implies, para, target neighboring cells. So, cell A secretes it, travels over to its neighbor to bind to a receptor on cell B. Very common example of this would be your immune response. So you have a T helper cell that releases a chemical and travels over here to a B cell to tell it, yeah, that's a bad guy, start making antibodies. That would be an example of a paracrine. Autocrine, on the other hand, is a hormone that is secreted by that cell and then targets that same cell. Okay, auto, self. So here, again, we can use T helper cells to say, you know what, not only am I sending a signal over to that B cell to say, yes, that's a bad guy, I'm also sending a signal to myself saying, yeah, that's a bad guy, I need more T cells to signal B cells. That would be an autocrine response. Now, chemically, hormones in general are classified in two ways. They're classified as either lipid-soluble or water-soluble. The solubility of a hormone has a variety of functions, or is important, I guess, for a variety of reasons. Solubility can determine how overall solubility. Has two main effects. Not only is it, of course, the chemical difference between the two, but overall solubility will actually affect hormone transport throughout the body. As well as receptor location. Okay, so these two things are determined by solubility. Okay, so let's take a look first at your lipid-soluble hormones. Lipid-soluble hormones are so named because they are lipid-soluble. Now, do you all recall from general biology what determines whether something is lipid-soluble or water-soluble? Well, hopefully, you're thinking polarity. So, remember, lipid-soluble hormones are non-polar. Okay? They're non-polar. Now, what does that mean? Well, that means they have a balanced arrangement of electrons. Now, that balanced arrangement of electrons results in all sides of the molecule having the same electrical balance. No one side slightly positive, one side slightly negative. Okay? Now, with that in mind, how does that affect the functionality of the hormone? Well, first of all, what is the blood predominantly made up of? Well, the blood is predominantly made up of water. As such, lipid-soluble hormones don't transport easily in the blood. They require a transport protein. What this transport protein does is bind to the majority of the hormone, making it temporarily water-soluble. This allows that lipid-soluble hormone to transport freely throughout the blood. Now, the majority of that hormone is bound to these transport proteins in the blood, and actually serves a couple of additional functions. In addition to making it temporarily water-soluble, it also makes it harder for that hormone to be lost via the kidneys. So by being bound to that transport protein, that lipid-soluble hormone sticks around in the blood a lot longer. It also creates a little bit of a mini reserve, so to speak, because since the majority of the hormones are bound to those transport proteins, they stick around longer in the blood, and you have a mini ready reserve. Now, obviously not all of the hormone can be bound to a transport protein, because then it couldn't it couldn't leave the blood to go to its target. So we do have a very small portion of the hormone freely floating in the blood, but that's referred to as your free fraction, and it is only a small amount of your lipid-soluble hormone. Well, we also said that that the solubility affected receptor location. And remember, we had two receptors, in general, two receptor locations. We had our plasma membrane receptor, and we had our intracellular receptor, okay, which is inside the cell. Well, and remember, we could either have a receptor in the nucleus or cytoplasm, or cytosol. Well, remember, if you're going to be bind to an intracellular receptor, you have to be able to get into the cell. Well, in order to get into the cell, you have to get through the cell membrane. Anybody remember what your cell membrane is made up of? Well, hopefully you are remembering that it is made up of phospholipids. It's a phospholipid bilayer. Well, if you're going to be able to get into a cell, you got to be able to go through the phospholipid bilayer, and the only hormones that would be able to do that would be our lipid-soluble hormones. So even though they have trouble transporting in the blood, they easily enter a cell to bind to intracellular receptors. Now, you'll want to review in your book all the different hormones that are considered lipid-soluble hormones, but in general, we have all steroid hormones, okay? Your thyroid hormones, T3, T4, are lipid-soluble, and finally, nitric oxide is a lipid-soluble hormone. Opposite of that would be water-soluble hormones. Water-soluble hormones include all of your amine hormones, protein-based hormones, and eicosanoids. Now, if lipid-soluble hormones are nonpolar, that must mean water-soluble hormones are polar. And this allows them to dissolve in water because, remember, water is also highly polar. Well, what does this mean? Well, since water is polar and blood is predominantly made up of water, then water-soluble hormones easily transport in the blood. They don't need any helpers. Okay, but since they are water-soluble, they cannot get through that phospholipid. So they cannot enter the cell easily. So they are stuck with cell membrane receptors. Okay, so we see that here. And of course, this whole comment about, chemical class determining transportation was addressed here in my lecture. Now, let's talk a little bit about the mechanisms of hormone action. Now, we saw in the nervous system that hormones, in general, ultimately resulted in a change in, with the nervous system, the neurotransmitter, resulted in a change in the permeability of, our cell membrane, typically causing some type of electrical signal. With hormones, the response is much more, or the potential response is much more diverse, and you typically have one of three possible responses. First, we're going to see a change in membrane permeability. Now, this change in membrane, oh boy, I sure spelled that one wrong. Let me go ahead and fix that. Let's try that again, shall we, folks? Permeability. Now, this change in permeability can result in changing in electrolyte movement like sodium and potassium, and it can result in an action potential or some type of electrical signal. But we can also change the membrane to the permeability of other molecules like glucose and amino acids. So this is actually one of the primary effects of insulin. It tells your cells to start picking up the excess glucose that's in the blood so it can be used inside the cell for a variety of different things. So the next potential function is a change in reaction rates. Now this can be either to speed up reactions or slow reactions down, but that change is going to result in typically the activation or deactivation of an enzyme. So we're either going to turn on or turn, just a minute. On or off some type of enzyme, because remember, enzymes are catalysts in our body and they speed up chemical reactions if they're present, and if we take them away, that chemical reaction would slow down. Okay. Finally, you can actually target gene expression. Again, either turning it on or off. Now remember, gene expression typically results in the formation of a new protein. Okay. Now this protein can serve a variety of functions in the body or in the cell, itself, resulting in a multitude of potential changes. Now recall, each target cell has the potential to have a different receptor type. And if they have a different potential receptor type, they can have a different potential response. This is what we saw when I mentioned briefly the idea that insulin at the liver is going to stimulate glycogen production, but insulin in your adipose tissue is going to stimulate triglyceride production. Insulin is insulin no matter where we are in the body, but the fact that you have a different receptor at those two different locations is what allows insulin to have different potential effects. Okay. And remember, we mentioned in the previous slide how receptor location will differ depending on the chemical class. Lipid soluble, lipid soluble is going to be intracellular receptors. Whereas water soluble is going to be a cell membrane receptor. So let's take a specific look at what happens when a hormone binds to its receptor. Okay, here we see an example of a lipid-soluble hormone binding to its receptor. Now remember, big differences here, and I'm actually going to go ahead and open up a blank screen so I can describe this here. So here we have our capillary, right? Key thing for the action Okay, key thing for the action of our lipid-soluble hormone. First, we, of course, in our blood are going to have our transport protein. That's not what I want. Okay, so we're going to have a variety of these hormones. Change our color here. So here we have our transport protein and our lipid-soluble hormones. Some are going to be bound, some are going to be free-floating. Okay? So this is going to be our transport protein. And this would be our lipid-soluble hormone. LSH is now my name for my lipid-soluble hormone. Now this would be the free fraction that's floating around. Majority, of course, is going to be bound to our transport protein. Now remember, for our lipid-soluble hormone, its receptors are going to be inside of the cell. So here we have our different receptors, different potential receptors, I should say. We have our cytoplasmic receptor, and we have our nuclear receptor. Okay. So what's going to happen is our hormone is going to diffuse from the blood through the interstitial fluid and head down and bind to one of two places. It's either going to bind, number one, to our cytosol receptor, or it's going to diffuse into the cell, down into the nucleus, binding to our nuclear receptor. That's option 2. So let's take a look at the potential responses here. So if, number 1, our lipid-soluble hormone binds to our cytosol receptor, it can have two potential responses. So 1A, okay, it will actually directly turn on an enzyme. It will either directly turn on or turn off an enzyme. This, of course, results in an either increase or decrease in reaction rates, depending on which one occurred, of course. Option 2 is the whole complex. So hormone plus receptor is transported into the nucleus. Where the response will be identical as if it bound directly to a nuclear receptor. So 1B would ultimately lead to the same result as if our lipid-soluble hormone bound to a nuclear receptor. Now, when our lipid-soluble hormone binds to our nuclear receptor, it's going to either turn on or turn off gene expression. Okay, so this difference, so we're going to ultimately either start or stop producing proteins. Oh, there we go. Okay, so this is our potential choices when a lipid-soluble hormone gets to its target location. Okay, is what we see here. If we go back to our previous slide, we can see that that's what's happening here. So here we have our transport protein, here we have our free hormone diffusing into the cell. Now this diagram only shows it binding to the nuclear receptor, changing gene transcription, new proteins result. Okay, but realize that I also went through what happens when a lipid-soluble hormone binds to a cytoplasmic receptor. So do make sure you're familiar with that as well. The tough one, of course, is the action of water-soluble hormones. Water-soluble hormones are much more complex because the hormone itself cannot cause the result because it can't get into the cell. So it has to use what's referred to as a second messenger system. So let's go ahead and take a look at it. Look at that here with another blank screen. Okay, so now we have our water-soluble hormone action. Okay, so again we have our blood capillary here and our cell down here. Here we're going to see our hormone. Since it's water soluble, it's freely floating in the blood, does not need any type of help at all. So it's going to freely diffuse from blood. But remember, since it's water soluble, it cannot get into the cell. It is blocked, so it is going to be forced to bind to our I'll use a different color so I try not to confuse y'all. It is going to be forced to bind to our cell membrane receptor. Okay, so our hormone leaves the blood, travels through, binding to our cell membrane receptor. Now our cell membrane receptor initiates a cascade of chemical reactions referred to as a second messenger cascade. So let's take a look at that in a little more detail. Okay. So here we have the water-soluble hormone, WSH, binds to Now, the example we're giving here is the cell membrane receptor is actually a type of G protein. Now, the G protein is the most common type of water-soluble hormone receptor, but it's not the only one, but it is the most common. So we're going to discuss it here. Your book does briefly mention some alternative response types for water-soluble hormone, but the most common is when a G protein is involved. So, first thing that happens is you activate the activated G protein, typically it's activated. The activated G protein is going to ultimately lead to the activation of an enzyme referred to as adenylate cyclase. We know this is an enzyme because it ends in ASE. Now this activated adenylate cyclase is used to convert ATP into cyclic AMP, okay, or CAMP. Now, cyclic AMP then becomes what's referred to as our second messenger. And this second messenger is released into the cell. Okay. Now, cyclic AMP then has the job of activating, typically, special enzymes called protein kinases. Now, protein kinases, which we are now going to refer to as PK, okay? Protein kinases add a phosphate group to proteins. Okay, well, why does this matter? What does this do? Well, remember back from general biology that proteins aren't just a string of amino acids, but they also have to be folded in a very specific manner. And if they're not folded in that specific manner, they're not going to function properly. Well, what do you think is going to happen to the folding of a protein when we start adding phosphate groups? Phosphate group being PO4. Okay? That shape is not going to stay the same, which means its function is not going to stay the same. So the added phosphate group by this protein kinase will either turn the protein, which is typically an enzyme, from on to off, or from off to on. Okay, and again, if we are targeting protein enzymes, we are ultimately changing reaction rates. Okay. Now, this is the typical response, but keep in mind, there are a couple of variations. For example, the activated G protein can actually deactivate adenylate cyclase. We can actually decrease the concentration of cyclic AMP as well. So there are a little bit of variations, but this is probably the most common second messenger cascade that you'll run across. A couple of quick questions though. Notice we said that cyclic AMP was the second messenger. Anybody here can you all think of the first messenger? Well, hopefully you all are thinking about this water-soluble hormone, because the water-soluble hormone was the first messenger. But since it couldn't get into the cell, it had to create a second messenger to finish the job. Now, remembering what we had talked about with hormones being able to work in very low concentration, well, here I'm going to demonstrate why that is. Okay, so we take, and let's try some green now. Let's say we have a one water-soluble hormone comes down and binds to our G protein, and it actually activates 10 adenylate cyclases. Well, each of those 10 adenylate cyclases then form 10 cyclic AMPs for a total of 100. And each of those 100 cyclic AMPs activate 10 protein kinases for a total of 1,000. And each of those 1,000 protein kinases are going to phosphorylate 10 proteins, leading to 10,000 activated or deactivated enzymes. So from one single hormone, we had an ultimate cellular response in the thousands. This is why we can see that hormones can work in very low concentration, and the reason being is the hormonal response is amplified. Okay, the hormonal response is amplified by each step causing a greater response, and so on and so forth. Now this is exactly what we see when we look at this response. So here's our G protein activating our adenylate cyclase. The adenylate cyclase forms CAMP. CAMP activates protein kinase. Protein kinase then phosphorylates our proteins. Remember, we are either turning it on or turning it off. Same principle. So let's go ahead and see what kind of result we've got with some questions. But first, some of you might be overwhelmed. Probably most of you, I'm guessing, and most of you are probably thinking, this is way too hard. She's making it harder than it has to be. Why do we have to know this? Well, first and foremost, wrong way. There are a variety of disorders that result when this cascade doesn't work properly. So if this protein kinase isn't working properly, or adenylate cyclase does not function normally, there's different treatments for those situations, but unless you understand this basic cascade, you're not going to understand how we would treat that type of a scenario. Not only that, this is actually quite simple, because this is your insulin signaling cascade. It's just a snapshot of your insulin signaling cascade. So if you thought that was difficult, this is what you could be learning. So I'm actually kind of making it easy. But this does demonstrate a couple of principles about how each receptor could ultimately activate a different type, or could ultimately stimulate a different response. And here we see a great example by all the different potential protein kinases. Each of these PKs and even AKs is a protein kinase, and each of those protein kinases results in a different potential response, okay, that we see here. But now let's go ahead and take a look at some questions to see how well you guys have understood the previous material. Okay, so reviewing again what we've come up with, downregulation makes a target cell less sensitive to a hormone. Well, if you're downregulating, you're decreasing the number of receptors, and if there's fewer receptors, then there's fewer places for that hormone to bind, so that hormone is going to have less of an effect. So this is a true statement. So, which chemical class of hormones is not matched correctly? So take a second, read this, and pause your recording while you come up with an answer. So, nitric oxide, lipid-soluble steroid, lipid antidiuretic, lipid, T3, T4, water, catecholamines, water. Not matched correctly. Well, what do we come up with? The answer is D. T3, T4 is definitely water soluble. Now, for those of you who did not get D as the correct answer, you are not wrong, hopefully, because there is actually a second correct answer here, and that is C, because C, antidiuretic, is actually water-soluble, protein-based hormone, whereas T3, T4 is going to be your lipid-based. When a lipid-soluble hormone binds to a cytosol receptor, which of the following is the first to occur? First being the key. So pause the recording and come up with an answer. So with the first to occur and a cytosol receptor, we have enzyme activity, activated, deactivated, hormone receptor moves to the nucleus, adenylate cyclase is activated, A and B are possible. Well, first thing to occur could be either A or B. So either one of those is possible. Again, read, pause if you need to, and we'll see what you come up with. So here it's water soluble, and again we're looking at first to occur. First to occur when a water-soluble hormone binds to its receptor. Well, remember, we activated a cascade of chemical reactions, but the first thing that occurs, according to what we learned, is adenylate cyclase is activated. Now, if this is activated, then this will be activated, and if this is activated, then that will be activated, and if this is activated, then that will be occur. So yes, all of those things will eventually occur, but the best answer here, of course, is C. So here we have a different type of question. So we're kind of taking it up a notch from simple memorization of some steps to understanding what is happening. So read this question carefully and see what you come up with. Pause if necessary. So here, if there was a genetic disorder that prevents adenylate cyclase from functioning normally, then, and again you're looking for the best answer. So, if adenylate cyclase did not function normally, well, really all of this question is asking is what's the function of adenylate cyclase? And the function of adenylate cyclase is to activate CAMP. So, if adenylate cyclase is not functioning properly, then CAMP is not being activated. Now, again, you can infer or assume some of these other things would also not occur, but the only thing you can guarantee is E. So here we have some more independent study. We had talked about the most common example for second messengers, which is the G protein second messenger system, and the use of CAMP as our second messenger. But there are other possible second messengers and other potential mechanisms. So I do want you to read through this on your own. And this is independent study. Finally, we have a variety of hormonal interactions that come into play when we want to look at the overall effects of or response to a particular hormone. Because remember, hormones do not exist in a vacuum. They have a variety of things. There are a variety of things going on in our body that could potentially affect what's happening. So, how much of a response we get and the overall intensity is going to be affected by a variety of things, including obviously the hormone concentration. The more hormone you have, the more of an effect you'll have, and so on and so forth. Again, along those same lines, we just reviewed it with downregulation and upregulation. The greater the receptor concentration, the greater the effect, and vice versa. But we also see that multiple hormones can interact and have different effects on one another as well. Starting first, actually I want to start down here at the bottom and work our way up. Antagonistic. Well, if you remember back to good old high school English, the antagonist in the story was opposed the hero or the protagonist. So antagonistic hormones, antagonistic effects and antagonistic hormones are when two hormones oppose one another. Best example of this, you actually learned about in AMP1. Calcitriol, calcitonin and parathyroid hormone are antagonistic hormones. Parathyroid hormone increases blood calcium levels, calcitonin decreases it. Another good example we'll talk about a little later is insulin and glucagon. Insulin decreases blood glucose levels and glucagon increases them. So those would be perfect examples of antagonistic effects. Well, next is synergy, synergistic effects. Well, when two things are in synergy, they are in unison, they are working together. Synergistic effects result when two hormones together produce a greater result than either one separate. So again, two hormones together produce a greater effect than either one separate. Finally, we have permissive effects. Permissive effects occurs when one hormone permits a second hormone to function, but that first hormone does not actually accomplish the same task. Now, this is different from synergistic because the two hormones are working together in synergistic. They function towards the same goal. They both stimulate bone growth, but in the presence, when they're both found together, they work to a greater degree. That's not the case in permissive effects. Best example I can give, if you ever watch those home improvement shows where when you're cutting really, really hard stones or tiles or things of that nature, you need a saw that often has water associated with it. Reason being is when that saw is cutting that very hard product, it creates a lot of friction and heat, which limits the functionality of that saw. So the water continually pours on it to reduce the heat and friction. But that water isn't actually cutting anything. It's simply permitting the saw to work with greater efficiency. That's the idea with permissive effects. Now, the best example I can give for hormones is the permissive interaction between T3, T4, your thyroid hormones, and catecholamines. Catecholamines like epinephrine and norepinephrine will upregulate the number of T3, T4 receptors on the heart. So if we have a greater number of T3, T4 receptors on the heart, reverse that, I apologize, folks. T3, T4 upregulate the number of catecholamine receptors. I'm very sorry for that confusion. I wish I could reverse and delete, but it's not that simple. So again, let me repeat. T3, T4 upregulates or increases the number of epinephrine, norepinephrine receptors, beta receptors on the heart, which means in the absence of T3, T4, you only have 5 beta receptors on the heart. Just throwing numbers out here, not being realistic, but just trying to give an example. But again, you only have 5 receptors on the heart, so your epinephrine, norepinephrine can only affect the heart 5 times. But in the presence of T3 and T4, now all of a sudden you have 20 beta receptors on the heart, and your epinephrine, norepinephrine can have 4 times the effect, binding to 20 receptors instead of 5. So, T3 and T4 permits your epinephrine or norepinephrine to have a greater impact on the heart, speeding it up, but it doesn't actually make the heart beat faster. Finally, how do we control hormone secretion? In general, hormone secretion is controlled in 3 ways. So we have control via other hormones. Okay, so we have inhibiting and releasing hormones. So you have growth hormone inhibiting hormone, growth hormone releasing hormone, thyroid stimulating hormone. So other hormones can control whether or not that guy is being released. The nervous system. So you can have direct signals from the nervous system that stimulate the release of hormones. And finally, chemical characteristics of the blood. So again, chemical characteristics of the blood. Now when I say chemical characteristics, you can look at it from a couple of different ways. Insulin's release is regulated by the amount of glucose in the blood, whereas the release of T3 and T4 is actually dependent on the amount of that hormone in the blood. So as T3, T4 levels drop, then we stimulate the release of more T3 and T4. Okay, whereas only when blood glucose levels rise do we get the release of insulin. So we see that here. Finally, as far as the actual control, 9 times out of 10, it's some kind of negative feedback loop. And as you recall, this occurs when the original stimulus is reversed. So too much T3 and T4 results in a decrease in production. Not enough T3 and T4 results in an increase in production, so we're reversing them. There are a few examples where positive feedbacks occur, but this is very rare in a normal individual, as positive feedback loops typically lead you further and further away from normal, which is not a good thing. We'll look at a few examples here, in particular those surrounding oxytocin and in childbirth as well as suckling, and we'll talk about those. But remember, for positive feedback loops, not a normal thing because it takes you further away from normal. Now recall, when we think about disorders associated with the endocrine system, most individuals focus on the gland itself and the idea that it's either not secreting enough, hyposecretion, or secreting too much, hypersecretion. Now in both of those cases, that is a very valid reason for a disorder to go, or for that hormone to be imbalanced. But one thing I want you guys to point out, and one thing I hope you guys are starting to see, is there's multiple things that have to happen in order for a hormone to take effect. Let's use a hormone, the thyroid hormone cascade that we just talked about, right? As you'll see here shortly, thyroid, the T3, T4 hormone release starts up in the hypothalamus with TRH. TRH heads to the anterior pituitary to release TSH. TSH then heads to the thyroid gland to release T3 and T4. Now, looking at this cascade, this is our goal, but a problem at any point along this signaling could lead to a problem. So it could be that we don't have enough TRH. It could be that we don't have enough TSH. Maybe we don't have enough TRH receptors here. Maybe we don't have enough TSH receptors here. Maybe your hypothalamus doesn't know that you don't have enough T3 and T4. Maybe the T3 and T4 are being produced, but they're abnormal. Maybe the T3, T4 receptors are abnormal. Maybe it's TSH that's abnormal. Maybe it's TRH that's abnormal. What I want you guys to really start trying to grasp here is it's not just, oh, the thyroid gland's broken. Hyper and hypothyroidism could be, in particular, because we're kind of talking hypothyroidism, you've got a dozen different steps in this process that need to occur in order for a T3 and T4 to get the job done. And a break in any one of those steps is going to lead to the same problem. The issue, of course, is the cure is going to be different depending on where the break occurred. So that is it for your first lecture. I know this was a long one. I'm going to try to keep the next couple shorter. I just kind of didn't have a good stopping point. So I will see you next time.